start streaming, it should be working. Let me check. It would come in about 10 seconds. Why can't you open, brother? Yes, we are live, but what the no, okay. We are live, but why isn't it showing here? I'm very confused about this thing. Why isn't live showing here? Yeah, it's getting shown in my laptop, but not really nice. Okay, let me just see. YouTube. Yeah, we are live. Just verify whether the sound's right. Yeah, we are live. A lot of background noise. Sounds right. Yeah, we are live. A lot of background noise. Sounds right. Yeah, we are live. A lot of background noise. Sounds right. Let me just check what if I do MacBook Air microphone. Now let me check. MacBook Air microphone. Now let me check. This is better. MacBook Air microphone. Now let me check. This is better. I need to check it in my phone because hello, I'm checking this. Yeah, this sounds right. Okay, welcome to day 2 of learning, understanding cancer, specific type of cancer in our case. And this is the screen again. Yeah, not this, but I think I can just close this. Yeah, so just to give myself a context of what happened yesterday. After this stream, I was like, I should be able to find something that I can improve upon from this paper. It can't be, the presentation can't be the same thing as what it is. So I just made a simple Google search of, okay, just to give more context. We decided upon CAR-M therapy plus a PD-1 inhibitor, right? Right. So it's anti-PD-1 plus CAR-M therapy, and CAR-M therapy very difficult to change because there's already set of protocol which is already done. It's already done. Okay. Should be working now. The display capture hadn't been reset. Just wait for some. Yeah, it's working now. Good. Just to give you a context, we had decided upon CAR-M therapy and anti-PD-1. So combination of these two. And I just tried to ask Google what are the different APD1s that we could use. There were three recommendations. One is a different antibody. So it's called as monoclonal antibody. The one which we already studied yesterday, I mean which they have used is also a monoclonal antibody, which is this. Pembrolizumab is a monoclonal antibody. Combination with CAR-M works good. That is what they have done. I tried, okay, CAR-M therapy is too difficult to change. What if we could change something related to the anti PD1, basically PD1 blockage? So then I was like, okay, this is a this is a good start to go to. Then I went to Google, as I said, and I asked what are the different blockade methods that we could use. So one is MAB. There are different MABs, which I thought, okay, this is very basic. Next, I saw something called as small molecules. Basically, some molecules that again either block PD1 or PDL1 or sometimes both. Which again, I thought, I mean, there is nothing involved for me to do, for me to create that excitement in me. Then I saw there is something called as DNA aptamer, and I am slightly exposed to that concept because of another project. And I am. So, just to give you an understanding, DNA aptamer is a short sequence of DNA, short sequence of nucleotide, and that can form any shape that we can give it to, if I'm sounding right. So basically, we can use DNA, a strand of DNA, to basically go and block PD1 or PDL1. And I tried to search a paper. We did get a paper. We did get a paper, and the paper is hopefully here. Okay, I have to open it. The paper is in. It is Immunology Against Cancer Papers, and this is the paper that I was talking about. Yeah, so this is the paper, and I mean, I did search 3-4 papers, but this one is a paper where they are using DNA aptamer targeting PDL1, which we want, and they're also working on the tumor model that we have already results on, which is the colon cancer, which we are working on. It's the exact same thing. Where is it? Yeah, it's the same cancer. Where is it? Where is it? Which is it? Okay, they're talking about aptamer at this point of time. PDL1 antagonist ID and aptamer. Okay. Oh man. Where did I study yesterday? That this paper was related to. It should be there in. Yeah. The results suggested that APT PDL1, which is aptamer PDL1, which they have created accumulated in PDL1, which is the ligand that is expressed by the tumor, which we saw yesterday, expressing CT26 tumor tissues, where it might interrupt the interaction between PDL1 and PD1. Now, CT26 tumor tissues are the same thing. It is colon tumor tissues, I think. Just to be a little, we'll just verify this once more. Which is the other one? This one. Find CT26. It is the same cancer. It is colorectal cancer, which we saw. Yeah, it is a general terminology for colon or rectal cancer. So they are also using the CT26 cancer model, and here these people are also using the CT26 model. So I was like, good. So my objective is very simple. Now I have got two papers working on CT26 colon cancer, working on overexpression of HER2, if I'm not wrong. Yeah, I think I'll find that one also. I think they have. Not really. Oh my god. Oh wow. Airspace 2. Okay, they're not working on HER2, but they're working on. This is a question that we should consider, whether this will play an impact. But they're working on the blockade mechanism between PD1 PDL1, which is good for us. So my objective is to do the CAR-M therapy as they have done in the previous paper, the older paper, this paper. Instead of doing this one, the treatment of MAB, I'll do the treatment of DNA aptamer. Now the thing is, in this paper they have just given DNA aptamer treatment. They have not done CAR-M therapy. So that is the novelty which we are trying to do. I did try to search whether they have done CAR-M and DNA aptamer. Apparently not. So this is a novel approach at this point of time, which we are going to stick to. So my presentation should look something like, we did this CAR-M therapy plus a anti-PD1. It worked like this. We did a particular DNA aptamer and it worked like this. Both of them had apparently good results. That's why they had there. I mean, the heading says so, that these are good results, anti-tumor effects. Our objective is to bring these two together. So our objective would be to do CAR-M therapy with APD1, and APD1, instead of an MAB, it will be a DNA antagonistic DNA aptamer. So that is the entire objective. We will try to read a bit more of the results and see how this goes. So at least we are clear on the idea what we are trying to do. So it's CAR-M. I mean, at this point of time, I can just take a paper which has CAR-M therapy. I think I searched, but I didn't get any. Let me go check it. It's Scholar CAR-M against CT26. They've done CAR-T. This is the one which we are studying now, I think. Yeah, it is the same thing. It is M-Ball 2022, no? Oh, it's not. Oh, wait a minute. M-Ball. Yeah, it is the same paper. 2022. Oh, no. Oh, okay. This is the further version of the same group or similar group, whatever it is. They had worked on that xenogenic mice. I think this is the paper that they're talking about. Xenogenic. Okay, there's something else only. But I think, yeah, this is the xenogenic mice model. And what we are looking into now is a more advanced one. Primary murine bone marrow derived macrophages were engineered. Okay, it is sounding the same. Or with syngeneic CAR-M monotherapy. Okay. I mean, this is a poster, so I assume after this they have. But this is a good poster for me to present my results as well, because if these are the results that they have presented in some poster presentation, it's easier for me also to. I'll just keep this downloaded because it's the same thing, but in a much shorter format. Right? Okay, we'll just go back. So this is just CAR T therapy. This is CAR M again, but this is 2020, and it's the same people. So it's one step behind, which we I don't think which we want. This again 2021. It's the same people. M ball, CAR T. I don't need. Okay, we don't have apparently. We don't have. Is this who is this paper? S Lee. S Lie. Oh, this one. S Lie. Okay, this is a different paper though. Engineer membrane. Okay, they have done something. What have they done? Okay, they are working on HER2. We again studied what HER2 is. HER2 is a factor which is mostly increased and most likely in breast cancer, but we are using that only to increase the tumor growth in CT26 in our case. Okay, that's not a thing to be worried about. CAR CT, I have no idea which is. Okay, CAR M. Okay, they are working on coated on to the liquid nanoparticle. Okay, CAR M liquid nanoparticle. Okay. Okay. Okay, these people are, man, nice. So they have done something similar to mine, which is, but I don't know if they're working with the same concept as mine of trying to blockade PD1, PDL1, because they have never not mentioned anything here. But what they are doing apparently is something similar to mine. Don't forget about the PD1, PDL1 blockade, but it is something similar. They are doing CAR T cell, but what is CAR M? It's not the same thing. I feel it's not macrophage that they are talking about. It is CT26 membrane. It is not. It is not macrophage. Okay, so I shouldn't worry about this one. It's not similar. Okay, just forget what I said. This is again M ball, and this is 2025. This is the one which we are looking into at this point of time. I think so. Preclinical models. Wow. So this is the only group that has been working in CAR M. Nice. Very nice. I mean, I assume this is very new then. Oh, good man. Good on me trying to find something which is very new. Let's just think for a moment. What if the idea that I'm proposing now leads to a Nobel Prize level, Nobel Prize level? I hope someone does this. If not, Probably I can do it as well. Let's see, I don't know. Let's see, I don't know. Okay, so we have no other papers other than these people working on CAR M therapy, at least against CT26. That is the one which I'm looking for because those are the relevant results, is what I feel. CAR M against a general cancer, is it necessary at this point of time? Is it a good control? So if I was just thinking about the narrative of how the presentation should be like. If it is, if it is, so we'll introduce to the people what CAR T cell therapy is. But I need to do it in 15 minutes, by the way. I need to show everything. To tell what CAR T, I'll just write it. I mean, I have, no, I'll write it down. What is today's date? Today is the 12th. Today is the 12th of March 26. So presentation. Probably order or something. What I was thinking is we'll tell that there is something called CAR T. Problems with CAR T. And this especially comes to solid tumors. It's not able to penetrate solid tumors because of fortress created. Um, why is my internet not it's very weird. If I do a window capture, let's check now. I should have done this. But it's not visible. No idea why. OBS, what are you doing? But it's not visible. No idea why. OBS, what are you doing? Okay. Print. So window capture is not the same thing. Okay, it's display capture, obviously, the entire thing. Okay, should work. That's what I did previously. I don't know why it hung. Now it's working. What if I do Canva? Is this also working? It's working. Okay. Okay, so if anyone has missed, I just thought of searching some papers relevant to CAR M against CT26, and apparently other than this particular group, which is MBall et al., there is no people who have done CAR M therapy. This is the same. This is the same group as well. M. Klinsky, MBall, and this is not CAR M. This is some other engineered cell membrane coated nanoparticle. So they are doing a different thing. They are doing LNP with mRNA. So different thing. But it's not, CAR M is not the CAR macrophage that we are talking about. And this is the same thing again, MBall, and that's it. I mean, I don't think anything will be relevant if it does not come in the first thing. Okay, this is a different. Yeah, this is a different. This thing they are doing CAR macrophages with liquid mRNA. Yeah, they are doing something. It's different. But as I thought, I don't think people have done the combination of a DNA aptamer with CAR M therapy, at least against uh, CT26, which is colorectal cancer. So yeah, so we can tell what is a CAR T cell therapy. We can tell what is the problem with CAR T cell therapy, and then we can introduce why CAR M therapy is better, right? We can then go ahead with the results of CAR M therapy probably. Um, yeah, we can go ahead with the results of CAR M therapy, and then we can give an introduction of CAR M. Then we can go for how can we connect this? So we can say about introduction to CAR M, and then we can say specifically that I found a paper where I had CAR M and treated with a monoclonal antibody, and we can show the results. Then I can say that we have found one more paper working with CT26, and they are using what? They are using the DNA aptamer, and they have found particular results. My objective is to bridge these two. The objective is the same. The objective is basically trying to increase the efficiency of the blockade, PD1 blockade, and using DNA aptamer instead of MAB, or a combination of both. Now I don't know whether a combination of MAB and DNA aptamer would be the right thing to do. We'll just do some basic research, basic search, whether it's possible. Can we use DNA aptamer and what else? MAB against cancer? You could use, and in combination. So we can probably design an experiment where we already have CAR M. Okay, we can do this one. Giving a rat CAR M, giving a rat MAB, giving a rat DNA aptamer to validate the results, which we have the results for. We can do combination of CAR M and MAB, CAR M and DNA aptamer, and we are hoping that this DNA aptamer plus CAR M, whatever we are done, is hopefully better than monoclonal antibody. And we can do one more thing, which is CAR M plus MAB plus DNA aptamer. Now I don't know what the issue is. I mean, first of all, I don't know if combination of CAR M and DNA aptamer is better compared to CAR M plus MAB. And if that is better, or even if that is not good enough, we can probably go with CAR M and MAB and DNA aptamer. But the only issue is MAB, both the MAB which they have mentioned plus the DNA aptamer are working towards the same thing. Yeah, this is what I meant when I said DNA aptamer. It's a short single-stranded DNA molecule that folds into 3D shapes, and it can be used to target the receptor. In our case, it is PD1. I think this is a this is a good start for the entire thing. I think this is the overview of the project, which is good. Yeah. So I just wrote, so CAR T, problems with CAR T, CAR M. We can share about CAR M. We can share about the paper CAR M and monoclonal antibody with the results. Yeah. And we can share about DNA aptamer separate results. And now what I'm thinking, my proposal. My proposal is a combination therapy of CAR M plus or minus CAR M plus or minus MAB plus or minus DNA aptamer. Makes sense? So we'll have just CAR M, just MAB, just DNA aptamer. We'll have combination of two each, obviously with CAR M. Or I mean, at this point of time, you can even have a combination of MAB and DNA aptamer, which they are saying it works. Or apparently, if, yeah, I mean, it makes sense why we should have CAR M at all scenarios. We should have CAR M at all scenarios because it will help sensitize the, yeah, that's what we learned, no? That with CAR M therapy, that's what we learned, I think. We'll go to the Canva. Oops. We'll go to Canva. This is what we learned with CAR M therapy. That there is a cancer cell, there is tumor associated antigen. It will get interacted with CAR of the macrophage, which we have engineered. And then this leads to phagocytosis. There is pro-inflammatory activation. There is inflammatory cytokines released, which will activate T cells. This will be done. In the meantime, we also have either MAB or DNA aptamer or a combination of both, trying to blockade the interaction between PD1, PDL1 of the tumor and other tumor, obviously other tumor cells, PDL1 and PD1. So basically trying to avoid the entire thing. What are the expected results? Probably it will be the same graph, but probably I'll just mark out that this is the result that we are expecting somewhere in this region. I think this is a good start. I had not planned anything today, but I think we can start with what do you say? We can start with the preparation of slides. But obviously, I'll not be able to share at least the template slides or the slides that I have from other people's slides on stream. So I'll do a copy of that particular template slide or do it somewhere else and probably stream tomorrow. I'll be free the entire day tomorrow. So I'll probably try to do as much as possible. Are we going on par with the schedule? Probably yes. Let me just see what I had written. Probably I'll just show you here. So this is what I had written. I had written this. Let me go to OBS camera. Is it ulta? Oops. Flip horizontal. Yeah. What is what to study for cancer, right? Okay, we are done with Wednesday 11th, we are done with Thursday 12th. We had to do CAR M and HER2. We had to do mind map and read papers. Completely, not completely, but it's fine at this point of time. And we had to prepare slides from Saturday. I'll prepone it to Friday and hopefully submit the presentation on before Sunday. That should be a good um, thing. Yeah, I think reading results would be much more easier because it's just oopsies would be much more easier than yesterday because it's just graphs and trying to understand graph. Let me give examples such that I become a little more confident in myself. It's not this, it's this. Okay, let's try to understand. This is something to do with MHC class 1 expression, which is not specifically important for us at this point of time. What is important is this. Yeah, we saw what is regional CARM yesterday. Regional CARM is basically the natural CARM that is there, and this one is the particular CARM. I mean, yeah, that is the cancer. This is the cancer. CT26 is colorectal cancer. HER2 is basically the method that they have used to induce colorectal cancer. So they have tried to increase the HER2 production, overexpression of HER2 in a mice, whatever that syngeneic mice is, and then they have tried to induce CAR T cell or try to induce CT26, and they are trying to see what are the different CARMs. Um, yeah, yeah, and what is the total tumor volume. So basically, they have induced tumor. This is day zero of tumor induction. Don't tell me that this was not visible. Oh my god. Grow up. Grow up. OBS. To do a display capture again. You know why it's sucking today. Yeah, I was just trying to explain this. Yeah, okay. This I was trying to explain. So this is the tumor volume in millimeter cube, and I was just saying what do we mean when we say CAR 26H HER2. So it is colorectal cancer induced by increased expression of HER2 against different CAR Ms. So one control that they have taken is untreated. They have not treated with anything. And this is the tumor volume. Which one? The green one. So it has gone till day 32, 31. Probably the mice has just died after this, right? So it's about, I don't know. It's high. If it would have survived, it would have probably increased more. The volume would have increased. UTDM, yesterday we saw that UTDM is basically normal macrophages injected. Okay, so it is also going high, but it is better than not treating it with macrophage at all. Then there is CAR M therapy. CAR M therapy is the same thing. Again, active recall. We'll try to understand what it is. You're taking monocytes, separating macrophages, engineering macrophages to have a CAR antibody on its surface. You're selecting particular things, a particular CAR M. Yeah, specific CAR Ms. You're trying to select it, making it in billions, re-injecting it to the mice or human, depending. So yeah, it is apparently good. So this is the CAR M result that we could show. That is, this is just normal CAR M, right? This is just, I mean, I'm just trying to figure out the And this is a better one. Okay, and this is again the same thing, percentage change in tumor volume till day 32. If it is untreated, what is the baseline though? If this 0 is the baseline, are they going in reverse? I don't know how. I think I should understand this better. Let me zoom it a bit. Obviously, I mean, percentage is going to negative, but I don't know what this actually means. We'll learn more. This one I know. This is a Kaplan-Meier survival curve or whatever. Basically, this is the number of days and this is the population survival, right? So 100% at whatever day 0, they had all mice surviving, and going on, this green color untreated dies after day 61 or sometime in 61. Population of mice which are untreated dies. With macrophages, there is some amount of survival, but the survival rate probability is very less compared to macrophage, CAR M. I mean, compared to the engineered macrophages. So, this clearly shows that CAR M is definitely better than M, completely better than untreated. So CAR-M is definitely a viable option, is what they have showed in this point, till this point. This is the combinational therapy that they have tried. This is again the tumor volume. This is again day 0 tumor. Everything starts with 0. They have injected something here. I mean, they have injected the tumor through HER2 expression. This is CAR-M. This is CAR-M plus anti-CD4, CAR-M plus anti-CD8. Just the macrophage, and CAR-M plus anti-CD8 plus anti-CD4. Apparently, treating CAR-M with anti-CD8 and anti-CD4, first of all, what is CD8 and CD4? Is it important? CD8 is the cytotoxic cell, T cell, and CD4 is cytotoxic helper T cell. Oh, okay, makes sense. They're doing something where they're trying to decrease the number or the activity of the helper T cell, decrease the activity of cytotoxic T cell, decrease the activity of both cytotoxic T cell and helper T cell. Why is that relevant to us? Very good question. This is a control that they have tried to do, but why have they done this control? Why have they done this control where they are trying to, what is CAR-M therapy? Let's try to understand. CAR-M therapy does not involve MHC at all. That we are sure of. Doesn't involve MHC. And MHC is required, I mean, MHC molecule attaches to, that's what we read here, right? This dendritic cell is nothing but macrophage, I mean, antigen presenting cell, sorry. Yeah, antigen presenting cell. And here is where I think I forgot. Wow. Okay. MHC class 1 molecules on tumor and MHC class 2 molecules also on tumor. If there is higher number of MHC class 1 molecules, it will enable the discovery of the tumor itself and most likely directly linked with CD8 plus cells, which is cytotoxic T cell. And for MHC class 2, it is CD4 plus cells. So yeah, so the objective in case of tumor cell is to decrease MHC molecules so that it decreases CD8 and CD4. So what they have tried to do is basically they have induced CAR-M and they have decreased the activity of MHC classes independent, I mean, indirectly. It's a good thing. Yeah, probably we'll read into the discussion later on. We'll try to understand why that is. Yeah, this is the mouse with colorectal cancer. CAR M they have given. Complete responders, 2 months post tumor injection, and naive mouse. Okay. Re-challenge with CT26 wild type, head to negative, okay. Tumor rejection, tumor growth, naive, okay, whatever. I think this is the method of how they have created tumor, and these are the mice that they are trying to grow, and this is how they are trying to work it out with T cell depletion with anti CD3. They are trying to reduce the T cells in the mice. Okay. Yeah, this is again naive mice versus CAR M therapy mice with tumor. So naive mice dies, every mice dies within 30 days, and CAR M everything survives. What is the difference between this and this? First of all, I need to understand what is naive mice. I need to understand what is naive mice. What is the difference between naive mice? Okay, I mean this is all mice having cancer, and think, what is naive mice? I think this is mice without any treatment. Tumor, naive mice with tumor, CT26 tumor, but no treatment at all, which I think untreated. So, what do they mean when they say this 100 to 500? Percentage change in tumor volume, day 31, relative to the baseline, 0, 100. Should I study it like this, or should I? How am I supposed to study this? In CAR M therapy, I can see that okay, the change is reverting, as in it's not trying, it's decreasing in size. But what is this one then? Why is this one getting decreased? I need to understand this better later on, or now probably, I don't know. Let's see. Yeah, this one, I think this is a good thing to study. It is the same thing, but with CAR M they have anti CD3. Why anti CD3? It is again like a control where they are trying to remove all the T cells, or kill or reduce functionality of T cells. We know that T cells are important to fight against cancer. So here, when the CAR M was given, the mice had all the T cells, but in this case they've tried to decrease functionality. Makes sense. Okay, and this is systemic CAR M versus 4T1. This is a different thing. This is not our cancer. This is something else. We need to see what this is. Yeah, we have the diagram description, so it's fine. We'll probably read it, it's fine. CAR M therapy inhibits tumor growth, improves overall survival, including long term anti-tumoral immunity. CD26 HER2 tumor was established SQ 14 days prior to treatment with IT. IT is intratumoral. I think so. UTDM. Okay. CAR M or left untreated to assess tumor burden. Okay. What I'm thinking, what I'm trying to understand is basically they have tried to induce CT26 type of cancer to the mice, to the mice 14 days prior to the treatment. Treatment can be anything. It can be untreated is also a type of treatment. CAR M is also a type of treatment. UTDM, which is normal macrophage, is also a treatment in our case, in this case. Normal macrophage is okay. Tumor growth curves are shown until the first mortality in respective groups and represent mean plus or minus sem. Okay, something mathematical. But okay, what I know is intratumoral, so they have allowed it to grow, the tumor, they have allowed the tumor to grow for 14 days. They have treated it with macrophage in um, nine mice. They have treated intratumorally with CAR M therapy in 9 mice and left untreated for 8 mice. So total how many? 24, 25 mice. Okay. Tumor growth curves are shown. Okay, shown till okay. Statistical analysis were performed two-way RM ANOVA in this one. A. This is what we saw. So, got it. It makes complete sense at this point of time. I think what they're trying to say, they injected it with. Okay. What is this? Okay, I mean, minus 14 is the day where they injected tumor, and day 0 is the treatment. I think day 0 is the treatment, and this is what they are measuring every time, right? Okay, we'll try to understand better. Waterfall plot shows the change in tumor volume on day 32 compared to the baseline. I did not understand this waterfall. What did he say? Change in tumor volume on 32, day 32 compared to baseline. On day 32 compared to baseline. On day 32, tumor volume on day 32 compared to baseline. First of all, on day 32 compared to baseline. 8, 1, 2, 3, 4, 5, 6, 7, 8, 9. I get it now. 1, 2, 3, 4, 5, 6, 7, 8. These are the different mice. This is 8 mice, this is 9 mice, this is 8 mice that we saw here. It's this one, n equal to 9, n equal to 8, and n equal to 8. So it's in different mice. So in untreated mice, relative to day 0, the growth has been 4000% increase. In the second mice it is whatever 3000, something like that, and in one of the mice it's just 150. Because in biological systems you can't just say every biological system has the same type of reaction against tumor. Depends on the number of T cells, depends on a lot of things. So it makes complete sense that they are taking each thing into consideration. UTDM also, I mean, yeah, I'm able to understand this very good improvement compared to yesterday. CAR-M. Now the thing is, see again, not all biological systems are the same. We can see that these 4 mice out of 8 mice have responded well. In fact, we can consider these two also. Tumor was 0 on day 0, slight increase in tumor, but this one is like complete increase in tumor. One mice hasn't responded well at all. Yeah, so probably, yeah, okay. Yeah, this is what they mean. So, how is treatment, how is the mice responding to the treatment is what they are trying to find out. And Kaplan-Meier curve over 118 days. 118 days is what, 4 months? Okay. Statistical significance was calculated using the logic log rank Mantel Cox test, which is probably one test. Graphs are representative of two experiments. Okay. Yeah. So untreated, they're talking about 8 mice. UTDM, they're talking about 9 mice, and this one is talking about 8 mice. So on an average, if we consider this is equal to 50, which is not equal, let's consider it's 50. At the end of 120 days or 118 days to be exact, 3 or 4 mice survived out of 8 in CAR-M therapy. This is the deaths happening. This is 1 death, 2 death, 3 death, 4 death, 5 death. Probably everything died. Yeah, everything died at the end of 62 days, 63 days in untreated mice. And probably one or two, a couple of them survived in UTDM, which is normal macrophage treatment. Yeah, okay, I was able to understand this. What about this? I think this I understood. Again, I'll see. Where are we? D, yeah, D. Where are we? D, yeah, D. Okay. CD tumor was established in the mice and were treated with for 3 consecutive days with anti-CD8, 7 mice, and anti-CD4, 6 mice. Combination of both, 7 days. Sorry, 7 mice starting on day 14. Starting on day 14. D. Starting on day 14. Somewhere here. When they say 14 days prior we injected them with cancer, are they saying that minus 14 somewhere here they started the cancer, I mean injected them with cancer, and on day this thing they are giving this particular treatment? I think so, right? Yeah, let's see. Yeah, IT CAR M, that is intratumoral CAR M therapy, or I think this is also intratumoral, was administered on day 17. CAR M for 6, UTD M for 7. 2A ANOVA, mean plus or minus 10. Okay. Okay. D, I was able to understand. Yeah, this I was able to understand. I mean, before also I was able to understand. I still need to understand how it is non-significant and significant. I mean, I think I need to do something to do with statistics a little more. Not really sure how they say this is. I've not worked with data. E. Okay, this is the next figure. We figured it out somehow, but I think I need to find out what naive is. At least what they mean. Schematic of post mice CAR M therapy rechallenged with HER2 negative. Oh, they tried to. So they had already given CAR M therapy for 3 mice. They rechallenged it with the CT26 for 2 months post com. What is CR? What is CR? Come on, brother. No, don't be like this. I wonder what is CR. What is CR? There's no CR. Wow. Here, I mean, come on, you didn't mark this one. Wow. Don't tell me this got stuck again. OBS, idiot. Properties, display capture, window capture, and back to display capture. Now it should work. Oh, not this. This. Hmm. Hmm. I should probably check. Okay, we'll see. We'll figure it out later. My OBS is giving some hard time today. Yesterday it was fine though. I should probably restart my laptop. I don't know. I have a bad habit of not shutting down my laptop. It's probably needed if I'm doing something heavy like this. Ah, OBS got stuck again. Wow. Restart capture. It's working now. Fed up with this. Okay, yeah, we were here, so I was able to understand this, and now we were doing. I can go full screen on this. Yeah, now we can go for F. Kaplan-Meier curve over 40 days post CAR M re-challenged. Okay, this is for the mice which is re-challenged with CAR M therapy. Naive mice were included. Okay. Yeah, so they're doing combinations of a lot of combinations they're trying to do. So they have increased the complexity. So it's not just giving CAR M therapy once. Post success, they are trying to induce mice with HER2 negative CAR M. No, sorry, HER2 negative tumor. And again, tumor grows, and then again, they are trying to do CAR M. That's what they are trying to do here. Yeah, again, the same thing. What is I? Three experiments, sir. Okay. Didn't really understand, but let's see. Okay, J is what? This one. G. What is G? Oh yeah, this we saw. H is 41C HER2 tumor treated with IV. Okay. This is treatment with intravenous CAR-M. 4. This is a different type of tumor. What is this? First of all, what is there? There are many questions at this point of time. I need to mark. First of all, yeah, and this one. T1. Yeah, CHER2 is basically trying to increase, but what is T1 type of tumor? I hope this one doesn't get blasted at this point of time. T1 tumor. Ah, it's breast cancer. Oh, what? Oh, no. Okay, wait. T1 C HER2 tumor. Let's try to search the same. C HER2 tumor. T1 is early stage tumor that is considered clinically HER2 positive. Okay. But what type of cancer it is? I mean, at this point of time, we know that HER2 increase can lead to cancer. I mean, what model is this? Oh wow, something else came now. Wow, I have no idea. Oh. Wait, we'll try to see there. First of all, okay, I understood what is T1. T1 is a early stage cancer. They are trying to re-induce post-CAR M. Which is this one? This one is H, which is this. Okay, this is a different one. This is a different one. Okay, this is a different cancer model. Let's see what this is exactly. If I can understand. Come on, brother, what is this? And what is orthotopic? Ortho, that is also 4T1 breast cancer. Oh, 4T1 itself is it's not 4 number of mice. It is 4T1 tumor. It is 4T1 tumor, but I don't know what is this specifically. Where is this tumor located? Parts of mice. top view. Parts of mice top view. Why do I need this? Because I need to see. Oh wow. Because I need to see lab. Wow, what is this? I mean, I need something. There's a warning. Okay, I need to. I don't know. I don't want this to get any type of strike because I'm showing something like this. This colon. Where is colon? Colon is what's something to do with large intestine. No. Let's see. Basic questions. Colorectal cancer in mice. Where do we get it, brother? Where do we get it? Oh man, show me the. Yeah, this one. So it's somewhere here in the bottom part and in the center. Is this colorectal cancer or is it breast cancer? That I'm trying to identify. What is a breast cancer like? Oh, breast cancer is in the top and also in the dorsal part, I believe. Yeah, it's some. Okay, that's a different. Oh wow. I don't know. I don't want my channel to get demonetized or anything. Yeah, okay, this is breast cancer, guys. This is breast cancer. So this is definitely not breast cancer, is what I think. This is not breast cancer. This is fluorescence. What is this? What is this cancer? He has not even mentioned it anywhere. Or have I not studied it well? I can't blame on them. Let me search here. OBS has crashed again. Congrats to OBS for doing this again and again. At this point of time, I need to figure out what's the problem. Okay. I think, yeah, I'll be done in a couple of minutes. Not a couple of minutes, but in some time. Yeah, what is this? Human breast cancer. Okay. Breast cancer. Oh, this was in the previous page. Okay. I did not read. Sorry, my mistake. Okay. Yeah, if we are looking into the model of CT26, this is not really important if we are trying to propose it specifically for CT26 type of. Okay, anyways. Okay. And what does orthotopically mean? What does that mean? Do we have that? Orthotopically. Yeah, this is the first one. What is this one? Well, orthotopically implanted in mammary fat pad, meaning breast. So they have not used anything like the previous one. CD26, they have not implanted, if I am right. I remember CT26. How did they induce CT26? I mean, how did they get colorectal cancer in this mice? Yeah, this the preparation of mice wala part CT26. Ah, okay, we had studied this yesterday. I forgot. 41 is the breast cancer. Okay, this is what. Okay, they have worked on both. Oh, hey. Okay, I wanted to see how they have tried to induce CT26. It's not really important for me to go for mice preparation at this point of time. Mm, yeah, it's not. It's really not necessary. Mice preparation is not necessary. Mice preparation is not necessary, and if I'm trying to work on a particular mice model, I want it to be CT26 because I have the results for DNA aptamer on CT26 model, mice model. Okay, this is what we studied now. Yeah, so this is the and ortho. What is it? Orthotopically. This is what it means. Injecting cancer. Corresponding to organ of origin in animal models. Okay, and what is the other method that they have tried to do? No, it is colon cancer. But how did these people induce this one? Is it also genetic modification to make it relevant target? Yeah, syngeneic system used to, yeah, immunocompetent, which is higher immunity, like normal immunity. HER2 expression must be induced through genetic modification. Okay. So there is CT26 wild type and CT26 HER2. Okay, I think so. When they say this is CT26 wild type, CT26 wild type, CT26 HER2. This is the engineered one. This is the one which mimics. What is the difference between wild type and SC is subcutaneously. Okay. Implantation is subcutaneous in this one. Have they mentioned SC anywhere? Find out. SC. No, not this one, brother. Not this one. No, here. Or S dot C. No, S dot C dot. No, it's not this one. Okay, it's not. It's not this. Okay. Anyways. Yeah. Okay. And what have these people tried to work on? These people. Is it CT26 wild type or is it HER2? This one is probably on wild type. That is the difference. Okay. We don't have the exact thing. Okay. How is it working perfectly? It's not. Okay. Yeah, it's a similar graph. Okay, I assume they're doing wild type. They're not doing it with respect to HER2. Yeah. I think okay. In my proposal, HER2 is not significant because we are not using it in both. HER2 is not significant, but HER2 is needed for. I mean, they have done it because it's more closer to the human being cancer, specific cancer. Okay, HER2 induction. Yeah, this is the tumor that they have gotten. Tumor size with anti-PD-L1, aptamer PDL1. Okay. Yeah, tumor is significant. Okay. HER2 is of very less significance. Okay, no worries, no worries. Yeah, I think I'll stop for today and we'll prepare slides tomorrow. Yeah, we are going somewhere. It's a positive thing. Mm, there is some clarity also that we shouldn't go for HER2. HER2 is not a very. I mean, at least at this point of time, we can't compare HER2 because we could design an experiment for HER2 CT26H HER2. We could do that. We could do that as well. Yeah. What is the other one? It's this. Okay. Yeah, so should be good for left hand. It's right hand here. I think I should change. Flip it horizontally. Yeah, that's fine. Yeah, yeah. We are done for today, and we'll prepare slides tomorrow. We'll start preparing slides tomorrow. Yeah, should be good. It's a 85-minute stream today, and there were some problems with OBS. I'll see what to do with it. So, see you and good night. Bye.